All right. Hello, everybody, and welcome to the workshop on fundamental of kinetic modeling. Um, we are very excited. This is the first workshop that is being set up by the NIF Pet Fellow Networks, and I'm pretty sure there will be more work coming in the near future. Without wasting more time, I'll just hand it over to Professor Stephen Meikle, who kindly accepted to give us a lecture and tutorials on the fundamental of kinetic modeling. Thank you very much, Stephen. We're very excited to get it started.
No problem. Thanks, Yasser. Yeah, look, welcome everybody. This is just to give you a little bit of context. This is a tutorial that I ran for a short course that we ran as through the IEEE, the Institute for Electrical and Electronics Engineers, IEEE, about 2 months ago in Vietnam for a class of medical physics and medical engineering students at different sort of levels, some undergraduate, a number of graduate students, and some people that are already working in the field. So it's a very introductory course, and for those of you who already have experience with using kinetic modeling tools in PET, this might be a bit basic for you, but hopefully you'll still pick up something new. I usually pick up something even when somebody's taking me through the basics of kinetic modeling. And for those that haven't done any kinetic modeling before, yeah, the aim really is to just give you enough of an introduction to the main concepts that you can pick it up from here and get into more advanced methods later. There's a little bit of lecture material as well. So when I ran this in Vietnam, I gave a lecture in the morning for about one hour, and then the tutorial exercise in the afternoon. So I'm gonna, for this session, for the next hour and a half, I'm going to give you a little bit of a hybrid. It's going to be a little bit of both. I'm not going to give you the full lecture, just snippets from that lecture that are closely related to the tasks we're going to do in the exercise. All right, so I'm assuming everybody can see the website that Yas has set up on the NIF website. So I'm just scrolling down a little bit, and I just want to remind you all, if you haven't already, click on this link here, Colab link, and that should open up this page. You should have that open in your browser. This should work in any browser on any machine. We're going to be using compute resources that are allocated in the cloud by Google, so it doesn't need any particular resourcing on your own computer. So just do that if you haven't already. And now going back to the website, the other thing, just a reminder, if you haven't already done this, to click on these links as well. There's 4 text files here that contain PET time activity curve data. We're going to be using maybe not all 4 of these, but some of these data files during the tutorial. So if you click on those and just download them to any directory on your local system. So you can just leave them in the downloads folder or wherever, but just remember where you put them. So you should have those stored locally. And then before I start with the slides, I'll get you to do just one more thing. And that is go to the Collab notebook. Here's a description of the tasks that we're going to run through in the tutorial. So I won't go over those just yet. That'll become clear once we go through the slides. But what I'll get you to do is just click on the first code cell here. And what that will do is it'll allocate, you'll see in the bottom right-hand corner, it'll tell you that it's connecting to Python 3, and it might take 5 seconds or so, 10 seconds, but it'll allocate some CPUs to your session. So it's worth doing that just to save a bit of time. And can I just get a show of hands? Just do a thumbs up if everybody's at this stage already. If you've got the Colab open and you've downloaded the files and you've clicked on that first code cell. Okay, I'm seeing quite a few thumbs up. Perfect. Okay, if you haven't already done it, just work on that while I go through these first few slides. And you should be able to see my title slide now. Is that right, Yasser? Does that look all right? Good, perfect. All right, so I'm just going to show just a few slides to give a really gentle sort of introduction to the topic. As I said before, this is a lecture that I gave in Vietnam, so that's why you see that banner at the top there. So, as I'm sure you all know, one of the unique features of PET and nuclear medicine techniques in general is that we can capture the dynamics of traces that we inject, and there's some really rich information in that data. We often only take images, static images, after a fixed period of uptake, especially for clinical interpretation. But if we put the subject under the scanner right from the moment of the injection of the radiopharmaceutical for an hour or more and capture all of the distribution over time, we get some really rich dynamic information. So that's what this slide is showing, and this is show that again. Yeah, this is a time-lapse sequence from a dynamic study performed on our Siemens Quadra total body PET system. So yeah, there's some pretty useful information in those dynamics. Now, what we would like to do is model that changing distribution of tracer in the body. So I'll start off with just a really general model that can describe virtually any tracer that's injected into the body. So let's say we have a tracer, which I'm indicating by the red X on this slide. It's going to arrive via the arterial inflow side of the circulatory system, and it's going to exit via the venous outflow. And almost any small or large molecule that's labelled has a likelihood of being transported into the tissue space as it passes by. That could be an active process or it could be a passive process. So that first part of this slide is to indicate the tracer crossing into the tissue space. And that's a reversible process. Most tracers can go into the tissue space and they can exit the tissue space back into the vascular compartment. Now, from there's a number of different things that could happen to the tracer. It could become bound to a protein in the extracellular space, or it could become bound to a receptor that's on the cell surface or even inside the cell. So we'll just represent that by this box that says our tracer is now bound to some protein. And that could also be a reversible process, so it can become unbound and become free again. The other thing that can happen, of course, is that the tracer can become metabolized, which means that it changes chemical form in some way. So I've indicated that by this different moiety XP now. So I've still got the radiolabel attached, but it's a different chemical species. And that could potentially also be reversible. So it might go back to its original form, the so-called parent radiopharmaceutical, or it could just exit directly back to the blood, and in which case you'll end up with radiolabeled metabolites in the blood. So these are all the potential pathways, and of course it can get much more complicated than that, but this is just a general model to simplify all the possible pathways, or some of the possible pathways for a radiopharmaceutical. So what we want to try and do now is come up with a modeling framework that is more specific for our particular tracer that we're interested in. And we generally use compartmental models for this. And compartmental models can represent either a physical space or a pharmacological state. So, for example, the bound versus the free state, or it could represent a metabolic state. So for example, in that last part of that slide, we showed the tracer metabolized and the parent radionuclide. So these could all potentially be represented by compartments. And then we make a few fairly fundamental assumptions. These are assumptions we make in all our compartmental models in general. And then on top of this, there'll be some other more specific assumptions that we often make for different types of tracers. But the first common assumption is that the tracer the radiopharmaceutical is in a tracer concentration. So that means the concentration is very, very small, so small that it doesn't perturb the system that you're trying to study. Secondly, we assume compartments are homogeneous and well mixed at any point in time that we're taking measurements. The third is that we assume the system itself, the body that or the physiological system that we're studying, is in a steady state. So that means the chemical reactions and the physiological processes and end products are not changing over time. They've reached some steady state. That doesn't refer to the tracer, of course. The tracer is changing in concentration all the time, but the underlying physiological system is remaining in a steady state. So what that means is the rates of exchange between the compartments in our model are going to be constant, and that's why we call them rate constants. And then the final assumption is that if there are any labelled metabolites of our radiopharmaceutical, we assume that they're going to accumulate in some other organ that we're not the organ that we're studying. And they will, of course, potentially accumulate in the blood. So we might have to take that into account, but we assume that they don't re-enter the tissue that we're imaging. So they're just some of the common assumptions. And then I'm just going to show on this slide 3 very common compartmental models that we use in PET. In fact, 2 of these are ones that in fact, I think all 3 we're going to use in the tutorial. So the first one is the flow model that's often used to describe freely diffusible tracers for measuring blood flow. So we usually represent that by a single tissue compartment. So in these models, the tracer in the blood, in this case, the plasma component of the blood, we don't treat that as a compartment. You'll see in some of the pharmacological textbooks, they often regard that as a compartment in its own right. But in PET, we usually just refer to the tissue spaces as being the compartments. So that's a single tissue compartment model for describing a freely diffusible tracer. And of course, there are 2 rate constants. Capital K subscript 1, K1, describes the transfer of tracer from plasma into the tissue, and K2, the reverse transport out of the tissue back to the plasma. So the PET measurement, what we're measuring from our PET data, is the concentration of the tracer in tissue, which is whatever is represented by this compartment. The other common model is the 2-tissue compartment model for FDG. We use this 2-tissue compartment model for describing other tracers as well, but the really common one is FDG. It's a tracer that's used for a lot of different types of PET studies. So I'll talk a little bit more about this model later. And these are the key operational, or if you like, the key parameters on the right-hand side in the blue boxes here. So in the first case, we're mostly interested in K1, which gives us the flow. This is blood flow, this F. In the second case, we're more interested in the influx, the net influx rate constant, this K sub I. I'll explain that in a little while. And then the third type of model for a different class of tracer, these are receptor binding ligand radiopharmaceuticals, is this model, which you can see is, again, a 2-tissue compartment model, but now those compartments represent something different. They represent the radioligand in its 2 states, free and bound. And these rate constants, the K3 and K4 are the important ones that describe the rates at which the tracer becomes bound and comes off again. so the on and off rates. And K3 depends on the availability of receptor sites for that ligand. And the key parameter over here on the right-hand side is something we call the binding potential, BP. And I'll explain later on what this ND subscript refers to. It means non-displaceable, but we'll talk about that in a moment. And it's simply just the ratio of K3 to K4. So they're three really common models. We'll use those in the exercise as we go through the Google Colab. So what do these models represent? So here, let's just take the two tissue compartment model for FDG. And actually what we measure with PET, if we drew a region of interest on some part of the PET image, you'd get a change in concentration over time of the tracer in that region of interest. What you'd actually be measuring, or what we're trying to model, is what's happening in this combined tissue space. So this dashed blue box represents the signal coming from the PET, from the tissue in our PET image in that region of interest. So you can see that it represents the combined two compartments that we've described in our model. So it includes both the free FDG, the unmetabolized FDG, and also the metabolized or phosphorylated FDG. We can't distinguish between those two species with PET. We don't have chemical specificity to separate them. We just know what the total tissue pool is. So that's what the C subscript T represents. And of course, the CP represents the radiopharmaceutical concentration in the arterial blood as a function of time. And so we can get that in a number of different ways. We could take arterial blood samples throughout the PET scan, but that's a somewhat invasive thing to do. It's not that pleasant to have arterial blood samples taken. Alternatively, and especially on the systems that we have available now for both total body PET, but also most of our preclinical systems, it's pretty common that we have an arterial pool in the field of view when we're doing imaging. So we could draw a region of interest either in the left ventricle or in the aorta, and we could directly measure our arterial input function from that region of interest. Now, we actually, what we really want with our models is the plasma concentration, because it's the plasma that exchanges with the tissue space. So if we use this method, what we're generally assuming, even implicitly, is that the concentration of the tracer in whole blood, whole arterial blood, is the same as in plasma. So that's just an assumption that we're making there. And for a lot of tracers, that's true, but you need to be aware that that is an underlying assumption. And then the third approach is we could just measure the input function in a whole representative population or a representative sample of our patient population and calculate the average. So we might have done previous PET scans with arterial blood samples, and then we calculate the average. So if you use a reproducible method of injecting the tracer with a well-controlled bolus, and if you've got a tracer that has very reproducible kinetics in blood, that's a really appropriate method. You can calculate the population average input function. So that's what we're measuring, and that's the relationship to the models that we're using. All right, so I'm on the last slide before we get into the tutorial. So we're going to start off with a one tissue compartment model in our tutorial. So let's have a closer look at that one. As I said, this one is often used to describe freely diffusible traces. And in this model, K1, the rate constant K1 is equal to flow times the extraction fraction. And the extraction fraction is represented by this equation, which comes from the Renken-Krohn model. You can go and look that up if you like to learn more about what that model is all about. It's a very simple model that just describes the arterial vessel as a simple cylinder. So, so that's the extraction fraction, and it's unique to each PET tracer that we use. And it's described by this exponential term where you have the PS product, so that's the permeability surface area product, divided by flow. And there are two limiting cases for different types of tracers. So if we have a tracer that has a PS product that's much higher than the flow, you can see that gives you a K1, because the extraction ratio goes to 1, and so K1 becomes approximately equal to flow. So when we have a freely diffusible tracer like O15 water, the extraction fraction is very close to 1, and so therefore K1 is very close to blood flow. On the other hand, a lot of the other PET tracers we use, that's not the case, and flow is significantly bigger than the PS product. In that case, we say that it's a permeability limited or capacity limited tracer, and then K1 is more closely equal to the PS product. So yeah, that's just to show you two extreme sort of cases for different tracers. Now, with all of the models we look at, you can write down the steady state state equations that describe the rate of change of your tracer concentration in the tissue compartment. And it's just equal to, in this particular model, it's equal to what enters the tissue space, so that's K1 times the concentration in plasma, minus what leaves the tissue compartment, which is K2 times whatever the current concentration is in the tissue compartment. So that's your state equation. It's a first order differential equation. So it has a nice, neat exponential solution. And the solution to the model is this equation that describes our tissue concentration at any time t as a convolution between our arterial input function and this mono exponential, which we call the impulse response function. So when we do kinetic modeling, mostly what we're trying to do is to estimate the parameters here of the impulse response function. And once we've got those, we can make inferences about what's happening physiologically. So that's just a really gentle introduction to kinetic modeling in general and the one tissue compartment model specifically. And now we'll jump to the tutorial. I'll come back to this presentation a bit later on. So is all of that clear so far? I hope I haven't sort of got too far ahead. All good? Okay. So if you just switch to your Google Colab, you should have already, so these are the steps we're going to go through. So just in this first part, we're going to go through steps 1 and 2. So we're going to read some of that data that you downloaded. We're going to plot the curves, the CP and the CT tissue curve. And then we're going to perform nonlinear least squares fitting. Actually, before I do that, I'll just go back to my presentation. There was one more thing I wanted to show you, which is the parameter estimation part. So if these are the model parameters we're trying to estimate, K1 and K2, the way we do that is to use some kind of optimizer, typically nonlinear least squares, and we need to define a cost function. And usually the cost function is either the L2 or L1 norm. So it's the distance between our model predicted curve and our actual observed time activity curve. So this is our model predicted curve here. For any given K1 and K2, so long as we know the input function or we've measured it, we can then estimate or calculate what CT should be. So that's our model predicted curve. And we compare it with our actual measured curve and calculate the distance, which is our cost function. Then we iteratively update our model parameters, and we're trying to reduce that cost or the distance, if you like. So we're trying to minimize that cost function. So we might start with an initial guess for K1 and K2. So this is a surface plot for our K1 and K2. The green surface there is the actual cost function for a particular data set. And we start off with an estimate, calculate our cost, and then we use some kind of gradient descent or nonlinear least squares algorithm to find a better estimate. And we do that iteratively, and each time we update our cost function, we should be getting closer to the minimum, depending on what type of algorithm we're using. So we do that several times, several iterations, getting closer and closer to the minimum. Our cost function is getting smaller and smaller until you get to a point where it's really not changing much at all. And at that point, your parameter estimates are not changing much at all either. And at that point, you have some predefined criteria for stopping, and you say, okay, that's good enough. Those are my parameter estimates. So I just wanted to take you through that as well before we do the tutorial. Right, so we're going to do the first two steps. So the first thing, you should have already clicked on this code cell. That's just going to load a bunch of libraries that we're going to need. And the next thing you do is click on the next code cell, and it's going to bring up a prompt. So this is where you get to choose the file that we're going to load into our Colab. Start with this one, noisy FDG, not the really noisy. I've got some really noisy data if you want to play with that, but just start off with the so-called noisy FDG data. We'll open that, and then we'll plot it. So is everybody seeing the plot on my screen and on your own screens? Hopefully your plot looks the same as my plot. So there's our input function measured in blue. You can see it's a little bit noisy. It's not a perfect curve. And the orange symbols represent the tissue time activity curve measured at different time points, at different mid time points. And you can see it's somewhat noisy as well. PET measurements are always somewhat noisy. All right, so if you've already got that far, the next thing we're going to do is fit our model. There's actually a couple of code cells that, yeah, this one's just going to do a bit of interpolation of our data, so we'll just run that one. You've got to run each one of these in sequence for this to all work properly. So that's that code cell. There's a bit of hypertext here to it just shows you those same equations that I showed you previously on the slides. This next code cell defines the function that's going to calculate our model predicted curve. So for the one tissue compartment model, that's this one. It's going to calculate the residuals. And this one here is just what allows us to do the real-time plotting of what's going on. So let's just click that one. We still haven't done the curve fitting yet. That's just to set up the functions for doing the optimization and the plotting. The next thing we're going to do is enter some starting parameters. So I think your Colabs should already have these values in it, but if they don't, enter some values and click this code cell. So these are just my starting guesses for K1, K2. I'm also going to calculate a third parameter called the blood volume fraction. That's because when we look at our PET measurements, I said before that ROI measurement represents the tissue concentration of the tracer, but there is also some vascular component there, typically around 5 to 8% of what we measure in the brain is actually the blood in the tracer in the blood compartment. So that we're going to allow the model to take that into account. We're going to make that a free parameter and try and estimate that as well. All right, so now we're up to actually doing the curve fitting, the model fitting. So click on this next cell. This is where it's taking our starting parameters. That's where it's going to perform the least squares fitting. And this is where it takes those the parameter estimates and calculates our model predicted tissue curve. and it's calculating the residuals here. It's going to calculate something called the AIC, which I'll explain in a moment. And you can see the plots below it. So does everybody have these kind of plots, very similar looking plots on their screens? Just give me a thumbs up if that's working. Very good. Okay. So what this plot on the left is showing, we don't usually do this when we're doing kinetic modeling. Usually all we see is that final red curve, which is the final model predicted tissue curve from our optimized parameter estimates. But I got Francisco did the coding for this. Sorry, not Francisco. It was actually Alistair that did this for me. And I got him to plot what those curves look like after every second iteration so that you can see what it's doing. And remember I showed you the cost function with that green surface plot, and you saw the parameter estimates getting closer and closer to the minimum. Well, with each point on that plot, that's represented by the model predicted curve. So I think that's our very first estimate. So that's the starting parameters that we had, or it's probably just the second iteration after we gave it the starting parameters. So it's not very close at all to the data points. But you can see that with each iteration, or each second iteration, it's getting closer and closer. And then the solid red curve is where it ends up when we reach our stopping criteria. Now, what we've done here is to fit a one tissue compartment model to data which represents FDG, and I've already told you that FDG is most commonly modeled with a two tissue compartment model. So although it kind of goes through the data points, you can see it's really not doing a good job at all of describing the behavior of the tracer in this early part of the curve. And it's not doing a very good job of these later data points either. And so that's why we often look at the residuals. So the plot on the right is a plot of the differences between the two curves, the model predicted curve and the actual measurement. So if you like, this is our cost function measured as a function of time. Remember I said that, maybe I didn't say it, but in the code above, the cost function I'm using here is actually an L1 norm, which means the absolute difference between these two curves. So that's exactly what this is. It's the residual. So this is our cost function. But what you can see is, just like we saw over here, there's quite a lot of structure in this residual curve. What you want to see is data points in your cost function or in your residuals that are randomly distributed about 0. And that's not what we're seeing. We're seeing something that looks like there's a systematic bias in the early part. I can see quite a structure to that curve. Seems to have a minimum, a maximum, and then it comes down again towards the end of the 1 hour scanning period. So that tells me straight away that our model is doing a poor job of describing the data. And so let's just have a look at the parameter estimates. You click on that next code cell. Even though I can tell that this is a poor fit to the data, we can still go ahead and print out our parameter estimate. So that's our final model parameter estimate for K1, K2, and VB. You can see that VB actually hasn't changed from our starting estimate. And I know, I didn't tell you this, but the data you're processing is simulated data. So I know what the value should be, and that should not be 0.1. So we'll see later on what it should be. The other thing it's printing out here is something called AIC, which is the Akaike information criterion. This is a parameter that gives us an estimate of the goodness of fit in a single number. And the more negative it is, the better, or the smaller it is, the better. I won't go into the details of this, but it's an information criterion that tries to find a balance between the goodness of fit as measured by our cost function and the number of parameters in the model. So it kind of penalizes more. So you can make a very complex model that'll give you a beautiful fit, but you'll have a lot of parameters which are ill-defined. So this criterion tries to find a balance. It tries to penalize too many parameters. So yeah, it helps you decide whether one model is better than another by trading off the number of parameters or degrees of freedom versus the goodness of fit. Right, so I think I'll flick back to the slides now. The next thing we're going to do is try to fit that same data set with the two tissue compartment model. But before we do that, I'll just talk a little bit about FDG. So just to explain a little bit more about what's going on when we inject FDG for a PET scan. And this diagram, I think, shows quite nicely the biochemical or physiological pathways, if you like, for FDG. Actually, it's showing the glycolytic pathway. So if we had injected glucose, or if the patient was metabolizing glucose in their bloodstream, this shows the pathway that the glucose molecules would go through. And FDG is an analog of glucose, so it follows all the same pathways up to this point, but it doesn't go beyond this point. So whereas glucose gets phosphorylated and then goes into this pathway and forms pyruvate and eventually lactate, and it generates ATP in the process. FDG doesn't make that far. It stops at this step here, the phosphorylation step. This is how glucose generates energy for the cell. ATP is where the energy comes from. But FDG doesn't go that far. And so this is why, in general, we assume FDG gets trapped in the cell. It comes in, these are sort of unidirectional pathways, and then it just accumulates as FDG6-phosphate. And the two processes that dictate the rate at which it accumulates are the rate of glucose transport across the cell membrane and the activity of this enzyme called hexokinase. So that's the enzyme that regulates the rate of phosphorylation for both glucose and FDG. And it turns out that it's those two steps, glucose transport and hexokinase activity, that are enhanced or elevated, if you like, in cancer cells. And that's the reason that PET with FDG works so well for the detection and staging of cancerous tumours and for monitoring response to therapy. So all of these sites of increased FDG avidity that you see here are due to spread of the disease, the metastatic spread of the cancer. And that's the reason. It's because these processes are elevated. So if we go back to our two-tissue compartment model, we can now say something about what these rate constants actually represent. So the K1 represents that rate of glucose transport. So it's the glucose transporter molecule that dictates that rate of uptake into the tissue space. And K3 is the other important rate constant. It dictates the hexokinase activity. So it dictates the rate at which FDG is phosphorylated in tissue. And you'll notice that in this model, I've got no K4. Sometimes we do have a K4 and we allow the possibility that there might be a reverse process here, reverse phosphorylation. In certain tissues in the body, that actually does happen, and K4 is non-negligible. But a lot of the time, particularly in the brain, we assume that K4 is 0, and this is therefore an irreversible forward process. So the FDG just continues to accumulate in the tissue. And then this is the key parameter, the net influx rate constant, which calculates the net forward influx into the tissue. We call it the net influx rate in units of minutes to the -1. And it can be calculated once you estimate or know the value of K1, K2, and K3. Okay. Now, if we wanted to, and if we knew what the patient's blood glucose level was at the time they had the PET scan, we could then go ahead and calculate the metabolic rate of glucose. So that's more of a physiological measurement rather than a model parametric measurement. But to do that, we need to estimate Ki, we need to measure the glucose concentration in the blood at the time, and we also need to estimate this so-called lumped constant, which takes into account all the differences in behavior of glucose, the different metabolic rates of glucose and FDG. Usually, we don't bother with this extra step. The Ki is already a nice quantitative parameter that has direct physiological meaning. So so let's go back to our tutorial. We'll come back to the PATLAC plot. So that's just to introduce a little bit the model that we're going to try now. So we're going to do some least squares fitting, this time with that particular model, the two-tissue compartment model. The only difference is that in this model, I'm actually going to include K4. So I'm going to allow that to be non-zero, and it's going to be one of the model fit, one of the free parameters we're going to try and estimate. And so, yeah, this is showing you the state equations for the free compartment and the metabolized compartment, and the operational equation, which looks pretty complex, but yeah, it's not too difficult to code that up and estimate it in an operational function, a Python function. So that's what we've got here. So let's just click on that code cell, which is doing all the same things as we did for the one tissue compartment model, but that function is now implementing the two tissue compartment model. Our residual function is exactly the same as it was before, this guy here. And the plotting is the same. So we've done that. Next step is, so I've run that code cell. Next step is we're going to put in our model parameter starting estimates. So again, if you haven't already got values in these fields, you can enter those in the same as the ones I've got on the screen and click that code cell. So there's our starting estimates. And now we're ready to fit the model. So you can go ahead and click this code cell. And it's going to do the same thing as we did previously, but this time fitting the two tissue compartment model. And you can already see straight away that the final model fit looks a whole lot better than the one we got with the one tissue compartment model. It's doing a better job of accounting for that initial exchange. So this part of the curve is dominated by that first tissue compartment before it gets metabolized. The later part is dominated by the terminal compartment, and it's doing a better job there as well. And you can see, apart from that very early first minute or two of the dynamic, the residuals are now much more evenly spread around zero. So there's our zero. And they're more or less randomly distributed above and below that axis, that level. So so it looks like it's a pretty good fit, relatively unbiased. So I'm pretty happy with that model fit. A lot of the time when we're doing kinetic modeling, we might do this again. We might start with different starting parameters. We might not be happy with the fit we're getting, or you might just want to repeat it several times to see if you get the same parameter estimates, take an average and a standard deviation. You might want to look at the covariance matrix. There's lots of things you can do to really interrogate the parameter estimates you're getting. But we won't go into that in this tutorial. So I'm pretty happy with that model fit. So let's display the parameter estimates that we get. And you can see now we've got more parameters. So there's more degrees of freedom in this model. We've got a K1, K2, K3, and a K4. There's our VB again. And we've got a new value of the Akaike information criterion. And the first thing we notice is that our model, our optimization is telling us that in fact K4 is 0. I think we started with a non-zero. Yeah, that's a non-zero K4. It may look like a small number, but actually K4 is always a small number. And this, with a value like this, if this was the correct value, it would have quite a significant effect on the curve. So it doesn't, even though it looks small, it has a big impact. But actually our optimization is telling us, no, it's actually a 0 K4. And I can tell you that from when I simulated this data, it should be 0. So it's done a pretty good job of estimating that. And I can also tell you, I can't remember the exact values, but these are pretty close to the values that I simulated. I think it was very close to 0.105 or 0.11, something like that. This should be 0.15, so again, it's pretty close. And I can't remember, maybe K3 was 0.05 or 0.045, something like that. So anyway, I can tell you that it's done a good job. But usually you wouldn't know that. Usually we wouldn't be using simulated data. And so you really have to pay attention to the goodness of the fit, the residuals, have a look at the covariance matrix. And then you have a look at this AIC value. So you might remember that previously with the one tissue compartment model, it was more like minus 2,000. And now we're getting a value of minus 4,500. So that tells me that that is a better fit than the one tissue compartment model, even taking into account the fact that we've got increased degrees of freedom. Right, so that's a pretty typical way that we would use nonlinear optimization, nonlinear least squares, to do model fitting. We've tried two different models to fit to the same noisy data, a one tissue compartment model and a two tissue compartment model. We've done a little bit of analysis to just cast a critical eye on the results, have a look at the residuals, have a look at the AIC, and then you can make some conclusions. But that's a pretty typical way that you would analyze just one-dimensional time activity curves from your PET data. And you might do that for certain selected regions of interest that you're interested in. You might actually do it on every voxel in the image and generate a parametric image of these model parameters for each voxel. So that's a pretty common thing to do as well. So that brings us to the next part of the tutorial, which is PATLAC analysis. So I'll switch back to the slides. Is everybody still with me? Have I lost anybody yet? Okay, good. Thumbs up. That's good. All right. So we're going to just talk about a very common method of estimating that same parameter Ki for FDG, the net influx rate constant. And that's called the PATLAC plot, or to be fairer to the three people that all contributed to the development of this method, it's really PATLAC, Getty, Rutland plot. Rutland was a New Zealand nuclear medicine physicist who independently came up with this method when he was analysing renal curves, renal time activity curves. And Albert Getty is a Danish. I hope I've got that right. He's either Finnish or Danish. I think he's a Danish medical physicist. Kelly knows. Who also independently came up with this method at about the same time as Clifford PATLAC, who was a US physiologist, actually. Right, so there's our two tissue compartment FDG model, and we're gonna assume for the purpose of this plot that K4 is 0. That's actually a fundamental assumption of this method. So if K4 is 0, as I said before, the tracer is going to accumulate in that terminal tissue compartment. It's got nowhere to go other than into that compartment. If that's the case, then the tracer becomes irreversibly trapped in this second tissue compartment. Now, it's worth pointing out that with this PATLAC method, It doesn't have to be a 2-tissue compartment. It could be 3 or 4 or 100. All that matters is that the very last, the terminal tissue compartment, has a 0 reverse rate constant. So it's going to get, eventually the tracer will get trapped in the terminal compartment. But in the case of FDG, we usually just describe it in terms of the 2-tissue compartment model, and there's a 0 K4. If that's the case, we can now write down an integral equation, operational equation, that describes the tracer concentration at any point in time in terms of the integral of our plasma arterial input function with this coefficient. And there you see our parameter of interest out the front, the coefficient Ki. That's the one we want. And you have this additional term which describes the amount of tracer that's still in this exchangeable tissue compartment. So V sub 0 is the volume of distribution within this part of the model. And it's so this term is V0 times the concentration at any given time point in the arterial plasma. Now we can rearrange this equation and divide throughout by Cp, and we get this equation. So now we've got a ratio on the left, and we've got a single constant value. V sub 0 is a constant. So it's not dependent on time anymore. So now we've got an equation that's in the form of a simple linear regression, y equals mx plus b. So there's our y, there's our x, this is our regressor, and there's the slope m. So Ki is now the slope of a straight line plot, and V sub 0, the volume of distribution in this part of the model, is our intercept. And so that's what we do with the Patlack plot. We simply take our plasma arterial input function and we normalize it by the instantaneous measurement at a particular time, and that becomes our x-axis regressor, and we plot this ratio on the y-axis. So that's what it looks like. In fact, I think this is the exact same data we've just analysed. So there's our y-axis for the Patlack plot. There's what we sometimes call funny time or Patlack time or normalised time on the x-axis. What you notice is that apart from the first 6 or 8 data points, which are where the tracer is still equilibrating in that first tissue compartment, but after some point in time, it becomes a straight line plot. And that's the part we fit with a straight line. Just do a simple linear regression. So we ignore these data points that are not filled in. So the solid symbols are where I'm fitting the straight line to. And then my intercept is going to be equal to V sub 0, which we usually don't worry about too much for FDG, although it does have some physiological meaning. And the slope is Ki, which is the parameter we're usually more interested in. So that's a Patlack plot. It's a much simpler and more efficient way to get Ki. So if we don't care about the individual microparameters, this is a more efficient way to calculate the main parameter of interest, Ki. It's computationally very efficient. It's just a simple linear regression, which also means that you can do this on a voxel by voxel basis and estimate Ki for every voxel in the image pretty quickly and generate a parametric image. So that's Patlack. We'll go back to our tutorial. And by the way, if there are any questions, please go ahead and ask and stop me. I should have said this at the beginning, but stop me at any point. Put your hand up or put a question into the chat box. Yasser's keeping an eye on the chat box. Otherwise, I'll press on. So the next thing we're going to do is that exact type of analysis. So there's our Patlack equation, our integral equation. Oh, actually, sorry, that's the integral operational equation. This is the Patlak equation here, the transformation. So first thing we're going to do is enter the only user parameter we actually have to worry about, which is something called T star. So T star is the time, the time in terms of the actual PET dynamic time frame, after which I estimate that this is going to be a linear plot. So you usually have a pretty good idea, usually from about 10 minutes onwards, FDG becomes linear in most tissues. So I'm going to say my T star is 15 minutes. You can change that later on and replot it if you want to try different values. So let's enter that number by clicking the code cell, and then we'll click the next code cell. And this one, you can see there the very simple linear transformation that's going on. There's my integral right there of the arterial input function, and there's my transformation to give me the X and Y coordinates of my Patlak plot. Very, very simple. And then I'm just doing a very simple linear regression to get my slope and intercept. So it's pretty straightforward, and then I'm going to plot it. So let's click that code cell, and there's our Patlak plot. So I think that's the exact same plot I just showed you in the slide. If you want to, you can print out the slope and the intercept, the correlation coefficient, p-value, etc., whatever you like. And then right below the graph, I'm actually plotting or printing out not only the Ki value I got from this Patlak plot, which is that one, but also the value we got previously from fitting our two-tissue compartment model with nonlinear least squares. And you can see in this particular case, they're exactly the same. You don't expect them to be exactly the same. If I'd have gone to another significant figure, they would have been different. And sometimes they'll be more different than this, particularly if the data is very noisy. But this is not very noisy data, so we get pretty good agreement between the two methods. One other thing you might notice is that this slope value here is actually slightly different to this value here. The reason is that when you do a Patlak plot, it doesn't intrinsically take into account the blood volume fraction, that 5% or so that I mentioned earlier on. So all I've done here is to correct my slope for that 5%. That's how I got the 0.024. Right, so that's the Patlak plot. You would have, I'm sure you would have seen a lot of Patlak parametric images of Ki and read a lot of papers where they refer to Ki, and that's how you calculate it. So I hope that makes that clear. We're going to do something very similar with the next data set, the receptor binding tracer that we're going to analyze. We're going to do another type of linear transformation to analyze that data. So yeah, we'll see how that works in a minute. Just one more thing that I wanted to do before we look at that data set is I said right at the beginning of the slides that when we do this model fitting, in a way, what we're really doing is estimating the parameters of our impulse response function. Each of these models will have a unique impulse response. And what will differ is the exponential coefficients. Well, the exponential, if you like, the eigenvalues of that mono-exponential if it's a one-tissue compartment model, bi-exponential if it's a two-tissue compartment model. So what we're really estimating is the decay rates of those two exponentials and the coefficients of those two exponentials. So let's have a look at what that looks like, because now that we've done the fitting of our model, we've got estimates for all those coefficients and rate constants, so we can actually plot the impulse response function. That's what it is for that data set that we analysed using the two-tissue compartment model. And I just wanted to point out a couple of reasons why I find it interesting to have a look at the impulse response. We don't always do this. but it can be quite instructive. One of the reasons is you'll notice that the y-intercept is actually equal to our K1 value. So that's, you can do it's a bit of a sanity check to make sure you're getting a sensible value for K1. And if you scroll back, you'll see that when we fit the two tissue compartment model, we got a value of about 0.105. So that's looking pretty good. The other thing you can tell from this impulse response is that this constant, it's actually got, you can see it's a bi-exponential with a very rapid decay initially. So that initial rapid decay is coming from that first compartment, the rapidly exchangeable compartment. Then you've got this very slow terminal tissue compartment. And because K4 turned out to be 0, it actually plateaus out to a constant. So this is an exponential that has a decay rate of 0. And when you have that, it decays to a plateau value that's equal to our Ki. So that is the Ki value for this data set. And again, it's saying it's about just a little less than 0.025. And that's what we got, 0.024. So that's just a bit of a sanity check. And it's also a way of understanding what the impulse response function actually represents. The other neat thing is that you can do things with this impulse response. So if you calculate the impulse response, you can then say, because the impulse response is what this is, if you're like in an engineering description, you'd say, this is the response of the tissue to an input function that's a perfect bolus. So if we were able to deliver the tracer all in one go at the same time, so there's no spread out tracer, that would be an impulse or a bolus input, like a perfect bolus. And then this is our response to a perfect bolus. So that means we could take this impulse response function and convolve it with any type of input function we want to and see what the tissue response would look like. And it turns out that if you convolve that input function on the left with a constant infusion, so this is our tracer in the arterial blood, and we're now going to deliver it at a constant rate, or we'll deliver it in such a way that the concentration of the tracer in the arterial blood, the arterial plasma, remains constant throughout the whole PET scan. And if you do that and do the convolution, your tissue curve would look like this. And if you're observant, you've probably already noticed that this curve looks a lot like a Patlack plot. That's in fact really what a Patlack plot is doing. It's doing a transformation to approximate or describe what would happen if we had given a constant infusion. Because if you give a constant infusion to a system that has a terminal tissue compartment with no possibility for reverse, for efflux out of the tissue compartment, it's going to just continue to accumulate. And of course, once you get over that initial exchangeable, that component where it's reaching a steady state in that first compartment, once you get past that, the tracer will just continue to accumulate as the integral of this constant curve. That's why you get a straight line. So that's our Patlack plot. And if I had calculated the slope of that plot, well, it should come out to be Ki. But I think, yeah, you've got to be careful about the units and things to make sure it gives you the right result. So I hope that's a little bit instructive as well, just a little bit about the impulse response and how you interpret the Patlack plot. Before we go on to the next step, which is, yeah, we're now going to have a bit of a look. How am I going for time? Yes, about 20 minutes, a bit less than 20. I'll try and leave a little bit of time for questions if I can. Is that right? We made it an hour and a half, right? Yes, is that right? Yeah, okay. So the last part of the tutorial, we're going to talk about receptor binding PET tracers. And when you have a tracer that binds to a specific protein target, like a receptor, this is probably about as complex as it gets. We typically don't have more than 3 tissue compartments, and this is what they might look like. So you would typically have a compartment representing the free ligand, the free tracer. So in other words, it's not bound to anything at this stage. It's in the tissue, but it's not bound to anything. You would have a compartment representing your tracer, your ligand, bound to the protein that you're actually interested in. So this is your target protein or receptor, if you like. So we call that specific binding. That's specific. If we say it's specifically bound, that just means it's bound to the target protein. But then you also typically, almost always, will have your tracer binding to non-specific targets. So there could be plasma proteins. So this could be happening in the blood. It could be happening in the tissue. Your tracer is going to bind to off-target proteins. So other proteins, other receptors, plasma proteins, all sorts of things that are in the body. And this combined part, all of your tracer that's free and bound to your non-specific target, if you put a box around that part of the model, we call that the non-displaceable component of your tracer. The reason we call it that is because if I was to inject the patient with a drug that I know binds to my target protein, my target receptor, it would have no effect on what's happening in these pools here, because it's not interacting with the same protein. It would only affect the amount of tracer bound to my specific target protein. And we often do that. Sometimes we'll do a PET experiment and we'll either inject the patient before the PET scan with a drug that we know is in competition for the same receptor sites, or even during the PET scan, we might inject a competitor drug and try to displace our tracer from those specific binding sites. So now you can see we've got K1, K2, K3, and K4. These are the key ones that describe the on rate and the off rate, roughly. And we've got K5 and K6. But what we usually do, very often do with these tracers, is we make the assumption that these two compartments reach an equilibrium very, very rapidly, in which case we can collapse these two into a single compartment. And then I won't go through it. There's a lot of different variations on this model that typically become more and more simpler and have fewer parameters. Before I do that, I should just say that in this type of a model, the key parameter we're interested in is the total volume of distribution, which is essentially the ratio of our tissue concentration to the plasma concentration once it reaches an equilibrium. And that's calculated from K1 to 3 and 4 in this way. And I'll show you in a moment that if we can calculate or estimate this V sub T, volume of distribution, from that we can calculate the binding potential, which is the parameter I said at the beginning we're interested in. Oh, and there it is there, right? So, So the binding potential is equal to the ratio of these two rate constants, K3 and K4, at equilibrium. And that's equal to the difference between the total volume of distribution of this system and the volume of distribution just for this blue dashed box, which is our non-displaceable compartments. So it's the subtraction of those over the VND. Now, coming back to ND, I said right at the beginning that we have this subscript ND for non-displaceable. This is what it refers to. If I calculate the binding potential as this ratio for a system like this, where I've got a non-displaceable component. So, and that's always the case when we're doing an in vivo imaging experiment. Then we always use this subscript ND to indicate that it's the binding potential relative to a non-displaceable compartment. So that's what that's all about. But the binding potential is important because it tells it's proportional to the receptor density of the target protein or receptor that we're interested in. Okay, now we're going to go back to the tutorial in just a moment. We're actually going to do one more simplification, or really two more simplifications of that model. This is called the simplified reference tissue model, which is a very common model that's used for reversible receptor binding ligands in PET. The first thing is I'm going to make that assumption that there's a rapid equilibrium between my free and my specifically bound and my non-specifically bound compartment. So actually, I'm now assuming that these rates are also relatively rapid relative to the other rate constants. So if we do that, we can now collapse all of that into a single tissue compartment in our target region. But what I'm also going to do is I'm going to say, if I can identify a tissue, this is usually in the brain, a reference tissue where I know that there are no or minimal specific binding sites, then I can have a separate reference tissue compartment which contains only the free and the non-specific binding, but no specifically bound component. So then I'm going to have a K1 and a K2 for my target tissue, and a K1R and a K2R for my reference tissue. And so to show you what I mean by that, if I drew a region of interest around a part of the brain that I know is rich in my target receptors, the specific binding sites, such as the striatum for a dopaminergic tracer, then that ROI, the tissue time activity curve in that ROI represents this part of the model. And then I draw a separate region of interest around the cerebellum, which in the case of dopaminergic targets has virtually no specific binding. There's no dopaminergic neurons, or very few in the cerebellum. So I can use that as my reference tissue. If I do that, then my operational equation looks like this. And even though that looks quite complicated, actually it's not that complicated. You can use basis functions to calculate these exponentials here with different values of K2 and BPND. And then you end up with only three parameters you're trying to estimate: K2, BPND, and this one R1, which you can see here and here. So this operational curve, equation describes the target tissue time activity curve in terms of the reference tissue time activity curve and our model parameters. And so that's what we're going to do in the last step in the tutorial. I think that's, yeah, so this is what we're going to be doing in a moment, but I think I'll just leave that. We're going to do a Logan plot. So I think we'll do that in the tutorial instead of showing you in the slides. So, so let's just click on this cell code here, code cell. And it's going to give you another prompt to open up a curve. So let's open up falapride M3. So falapride is a dopaminergic tracer, binds to D2 and D3 receptors. And it binds pretty specifically to those receptors and not much else. And M3 refers, it's just the third mouse in a series that we did on our Brooker preclinical scanner. So let's open that. And there's the curves. So what I'm showing you here, and what you should have on your screens as well, is the target tissue time activity curve, which has come from, should just show you the regions of interest, I guess. So this is the actual animal. So there's our striatum, caudate putamen in the mouse. So that's our dopamine-rich target region, and there's our cerebellum there. These are just the eyes, or the hard-hearing glands behind the eyes, but there's the cerebellum, and these are the target tissues. And we're going to calculate this Logan plot. So going back to our tutorial, there's the target tissue time activity curve. You can see it almost reaches a plateau. This is a relatively slowly reversible tracer, but it is starting to reverse. And there's our cerebellum curve. This normally would come down a little more than this. I think there was just probably a problem with the way the atlas was fitted to the brain for this mouse, and it looks like it's actually slightly going up, which is not possible, but normally that cerebellum curve would be coming down a little bit there. The other thing I'm showing you on the right here is the ratio of those two curves. This is the striatum to cerebellum ratio. And I'm going to explain in a moment why that's a useful thing to look at. Just like the residuals were useful to have a look at before, this is quite handy. And I want you to just notice at the moment that this ratio curve reaches a plateau or kind of a peak at around about 6.5. Right, so the last thing we're going to do is compute this Logan plot. So you can see here from these equations that it's a lot like the Patlack plot. It's another linear transformation that's designed to turn our operational equation, this time for a reversible ligand binding tracer, into a linear regression. There's our y-axis. There's our regressor for the x-axis. There's our slope, and there's our intercept. But the interesting thing now is you see that we don't have Ki as our regression coefficient. It's the ratio that I spoke about just a moment ago between the total volume of distribution and the non-displaceable volume of distribution. Once we know that, we can calculate BPND. We call this the distribution volume ratio, DVR. And if you simply subtract 1, you get the BPND value. So let's go ahead and do that. We'll do another T star. So typically that's about 10 or 15 minutes as well for this tracer. And now the last code cell, we're going to do the Logan plot. And you can see once again, it's a very, very simple transformation. So there's our integral of our tissue time activity curve, target tissue. There's the reference tissue integral. And there's our Logan x-axis and our Logan y-axis. So it's a pretty, very, very simple thing to implement in Python. And there's our Logan plot. Now, the first thing I want you to notice, I've printed out the DVR estimate here. So remember, the DVR is the slope. If you look up here, the slope of our regression is about 6.66. So there it is there. And if that number sounds familiar, that's because it's approximately the same as this peak ratio value, or sort of plateau, the plateau value it's approaching. So that's a bit of another insanity check, just to make sure you've implemented the Logan plot correctly and you're getting sensible values. So our DVR is 6.66. If you want to go ahead and calculate the BPND, you simply subtract 1 and it's 5.66. So yeah, so that's a Logan plot. So you've now done a nonlinear least squares fit of a 1 tissue compartment and a 2 tissue compartment model to an FDG data set. We've done a Patlack plot, and we've then looked at another data set for a dopaminergic reversible receptor binding ligand called falipride in a mouse model, and done a Logan plot to calculate the binding potential. So we've got through a fair bit already. For the tutorial in Vietnam, I had a couple of questions here for the students to think about. We've already discussed why the DVR is similar to the cerebellum, striatum to cerebellum ratio. Maybe I didn't say that exactly, but that ratio at equilibrium for these types of tracers is actually just equal to that K3 to K4 ratio that I showed you earlier. So that's why it turns out to be the distribution volume ratio. Yeah, and a few other questions there. Now, there's about 7 minutes left. What we might do really quickly is just open up one of the other data sets. So if we go back to the prompt, so you go back to this code cell at line 21. and open up a different data set. So I'm going to open up mouse 6 now. And there's my curves. There's the target tissue curve, the reference tissue curve. So this is the striatum, this is the cerebellum. There's my ratio. You can see it's lower now. It's more like 5 instead of 6.5. And I'm going to redo my Logan plot. And this time I'm getting a BP that's close to 4.5. So once again, it's similar, not, oh, actually the DVR is 5.5. So yeah, it's pretty similar to the equilibrium value there of the striatum to cerebellum ratio. And you can see the BP is less for this mouse than it was for the first mouse. And the reason for that is that this mouse was treated with ethanol. So this mouse was pre-injected with ethanol, I think about 15 minutes before the scan or 20 minutes before the scan. And ethanol is a drug that causes a mild stimulation of endogenous dopamine release. So what's happening here is that the endogenous dopamine is now competing for our tracer for D2 binding sites in those dopamine-rich regions in the striatum, which means there's fewer receptor sites available for the tracer, so you get a lower BP value. And that's a very typical sort of receptor binding ligand experiment where you're testing some kind of drug intervention versus a control. The control animals had saline injected. So yeah, that's how we can use the Logan plot to estimate important physiological parameters and do some hypothesis testing in our animals. So I think that, yeah, that brings us to the end of the tutorial. I'll go back to the slides. That's the Logan. Yeah, the last slide here, I'll shoot these slides through to Yasser and we'll put them up on the NIF website if you like. That last slide has some references and some links that you can follow, a few nice reviews of different modeling, quite common modeling techniques. There's a couple of websites as well, the Turku Pet Center and PMod, the company that makes kinetic modeling software. They both have really nice resources for explaining a lot of the models that are used in PET. And I also really like this review by Roger Gunn and Julie Price and others that was published in PMB. It's 10 years old now, but it really goes into all the different, it's specific to the brain, it's specific to protein binding ligands, but it covers all the different types of PET traces, different categories of protein binding traces that we use, and the way we use kinetic models to derive physiologically relevant parameters. This one's a bit more this a bit more recent, and you can see this guy Meikle was involved. This was a review article that we published recently with Evan Morris and my former postdoc PhD student Gail and Jocelyn Hoy, who was one of Evan's postdocs. This one's much more niche. It's a very specific type of experimental problem in PET where we're doing drug challenge studies and we're trying to deliberately displace the tracer. So it talks about the different types of models, non-steady state models that are appropriate for those types of experiments. And then this one's more about total body PET kinetic modeling. Yeah, so I think that's it. Yep, that's it. So I'll stop sharing.
Thank you very much, Steve. That was a really great lecture and nice tutorial. I think we've got 3 minutes for any questions or any comments. I think you're getting a lot of love and appreciation, Steve. It was really
that's great.
enjoyable.
Yeah, more the better. So are there any questions there? There's some links there, which is great. Yeah, okay.
And I guess, if there is any questions, maybe, I don't know if they are welcome to email you, Steve.
Yeah, absolutely. We can put my contact details up on the NIF website as well if they're not already there.
Sure.
Yeah, happy to respond to emails if you've got specific questions. Yeah, that's fine.
Awesome. All right. Thank you very much, Steve, again, and thanks to others.
No questions?
No questions or comments or anything to discuss?
There's a question.
Yeah, would you be able to circulate the slide? Yeah, we'll do that for sure. We'll do that. We'll put it on the website.
Definitely do that. I'd also be interested in any feedback that you guys can give me about ways to improve this workshop. If there are people, I know there are people on this call that are very experienced kinetic modelers, much more experienced even than I am, like Kelly, for example. I don't know if she's still there. If some of you would like a more advanced version of this, we are working on another tutorial that we're going to run at the IEEE Medical Imaging Conference next month, which will focus on total body PET kinetic modeling. So, we could potentially run that at a later time, maybe sometime next year. But if anyone has any suggestions on other things they'd be interested, or, other topics or other models that I haven't explained that they'd like to see explained, we can do that as well. So, yeah, any feedback would be very welcome. I think that's it. Yes, no questions.
All right. Awesome. Thank you very much again. And
Welcome.
all right. Thanks a lot. See everybody.
Thanks, everybody, for your attention.
Thank you. Bye-bye.
Bye.
Bye.